and yesterday's training program, but I will now slightly move you to the textbook information as well as theoretical background of tree construction, different kind of algorithms, and different type of parameters that you will be visualizing during the tree construction. So let us start with the yesterday's example. I think all of you would have done multiple sequence alignment by now, and you are aware that what multiple sequence alignment means. So I will take the same example in a very layman's way, those who were not at all aware about phylogenetics earlier and those who do not know how molecular phylogenetics work. I will give you the same example of certain names. Like we have some names written over here: Amit Kumar, Sumit Kumar, Aditya Arya, Sneha Singh, Aditya Singh, Donald Trump. So these are some names which are given on the screen, and let us say these are not just names, but these are some sequences, and the letters in these names have relevance to their origin and to their diversification. If we are assuming this, let's say we keep some icons for recognition. Now, what I will do is, you have yesterday got some sequences during the practical session, and you might be aware that during the practical session, you got some letters of microbe sequences that were base pairs. They might be considered analogous to these names. And what you did in the next step was the multiple sequence alignment. So yesterday itself, I briefed you that what alignment means. You can align Amit Kumar and Sumit Kumar one over each other. Like you can write Amit, then Kumar, and you can write Sumit Kumar just below it in this manner. So if you do it like this and you try to match each of the letter, you are going to see mismatch everywhere. But if you do it in the other ways, like if you start S from here, if you shift the whole word one space on the left hand side, MITKUMAR will be matching there. So we learned that how multiple sequence alignment algorithms work, and they keep all those things in a optimal way so that you get the optimal score. I'm not using the word best score, because in order to get the best score, you might have to disturb the alignment to a great way, a great change. But we have to keep it optimal so that you get the best alignment which is relevant to the evolutionary significant. So let us just see. I have made some alignment, and you can see on the screen that how these names have been aligned here. So I just put names here, and these hyphens, they represent gap. Evolutionary point of view, on evolutionary point of view, these gaps may be representing deletion, or maybe if this name was ancestral, you can say there was an addition over this place. So indel are very common type of changes which happen during the evolutionary process, insertion and deletion. Now, what you observe during this whole alignment, when I did MSA or multiple sequence alignment, you will see that the names Amit Kumar and Sumit Kumar have something in common. Okay, so this part is common, and that becomes the basis of fact that I can keep these two names as one common family, or you can say they are connected somehow, and they can be kept together. They are likely to be originating from a common ancestor. So let's say I drawn this line in order to construct a relationship tree, and I kept number one, that is, let us say Amit Kumar here, and you can say if these two are related, then I can keep Sumit Kumar as a branch of this, because they are likely to have a common ancestor from which these two would have originated. Now, furthermore, if you look Aditya Arya name, you would realize that AR is common between these three. So I just made an alignment in a way that we get some common things. So since these three names have something common, it means Aditya Arya is also connected to these two names somehow. But it did not emerge directly from Amit Kumar. You can say there was a common ancestor to both Amit Kumar and Sumit Kumar from where Aditya Arya emerged. So I make another line here and I put this name over this place. Now, furthermore, if I look at the next name, Aditya Arya, Aditya Singh, then you will find that there is a lot of similarity between these two. So possibly Aditya Singh and Aditya Arya may be belonging to the same clan, and I can put that here. So this is representing Aditya Singh. You can relate with icons to recognize these structures. Now finally, you have name Sneha Singh, which is having some relationship with this name in terms of the similarity. And you can observe here that ASINGH, this is matching here. So since these two are also matching with each other and they are very closely matching, that means the fourth name, Sneha Singh, must be having a common ancestor here. So you can now relate that Aditya Singh and Sneha Singh, they had very large number of characters which were matching. So I placed them together in a family and they had a common ancestor. Then Aditya Arya and Aditya Singh, they had some common similarity and they were kept as a single common branching out partners. Since Amit Kumar, Sumit Kumar and Aditya, they had some common things like AR and IT in them. So they were also connected by a common ancestor. So this is how I constructed a tree. But now we will learn it later that we also need an out group which is not directly related with these things, so as to root this tree and so as to clarify the scale, or you can say clarify the level of emergence. So finally, I can say the Donald Trump, which is apparently not very much related, can be considered as an out group which is not a part of any of these. As you can see that no many letters are matching with the names. I have kept different letters with different names. So this can be considered as an outgroup. So this example will particularly illustrate you that how do we connect things, how do we find out similarity in the character-based methods. And if you create an analogy between the sequence and the protein sequence, you would realize that most of these letters, except U, are encoding for some of the amino acids. So you in reality can actually find out these kind of letters and protein sequences. And if you do protein-based phylogenetic analysis, this example will be very, very common. But right now we are talking about DNA-based analysis. So even if you have DNA-based analysis, you are going to have some similarity. And based on that similarity, you will be able to draw trees. So this is the fundamental of multiple sequence alignment and how that multiple sequence alignment is actually connected to phylogenetic analysis. Now let's move ahead. Let's talk about some of the textbook knowledge, what phylogenetic tree is. So tree is basically a diagram, or mathematicians will say that it's a graph. Okay, so it's not a tree that will have, that will do transpiration, that will do photosynthesis, but it is analogous to that because it will have a similar structure. It will have branches, it will have root, it will have shoots. That's why it is called as a tree. But mathematically, it is a graph that depicts the lines of evolutionary descents of different species, organisms, or genes from a common ancestor. So you should remember the fact that phylogenetic tree may not be representing the evolutionary descent of organisms, but sometimes it may also represent the evolutionary descent of genes itself, right? And it requires not only the knowledge of protein sequence, DNA sequence, but it also requires the knowledge of biological diversity. It also requires the knowledge of classification, phenotype, as well as evolutionary process. So you have to understand evolution, sequence, and all these things to understand how phylogenetic trees can be drawn and how do they work. Now let's look at some basic components of a tree as we have seen in previous slide. A tree will have some basic component. It might look like this, or it might have different shapes. So primarily a junction point from where the branching takes place, that particular point is known as node. Whereas there can be several nodes in a tree, And those nodes which are having only one branch and these are from the side of a root, they are the first nodes. They are called as external nodes. But if there is a branch coming out and there is another branch, then there is a node. That type of node is known as internal node. So we have nodes from where branching takes place. We might have external nodes and we might have internal nodes. Then these terminal points, they might be representing the terminal sequences or terminal species, and you call them mostly as leaves. Just like a plant, you know, in plants you have roots, you have then branches coming out, then you have more branches, and then finally you have leaves on them. So you can say these points are actually kind of nodes. Fine. And these leaves in technical terms can also be called as terminal taxons. Mathematicians will give different names. They will call these things as nodes and they will call these lines as edges, because as I mentioned that this is a graph. So graphs will have edges or vertices and nodes. Now, besides this, if you look at the specific parts of this tree, if I take this particular part shown in purple color, then this part is represented as paraphyletic group. What paraphyletic group means, paraphyletic group indicates that it includes ancestors of some descendants, but not all. So let me just first complete all the three types. This is polyphyletic group and this group is monophyletic group. So what you see in this particular monophyletic group, that if you have number 3, number 4 and number 5, they have all the possible ancestors represented here. So if you have a group where all the ancestors have been included, then that group is known as monophyletic group, or in other terms it is also known as clade. So studying such kind of trees where you include all the ancestors, that is also called as cladistics, where you talk about clades. But leaf number 1 and leaf number 3, they do not represent a same clade, because they have originated from different ancestors. And if you make a group of 1 and 3, probably you are not including the nodes. And in such situation, when you are not including ancestors, but you are only including the divergent species or divergent leaves, then that group is known as polyphyletic group. So maybe even if you include 1, 2, 3, 4 and 5 only, that particular group will also be called as polyphyletic group. But here, what paraphyletic means? Paraphyletic means you are including a organism where all the ancestors have not been included, or it's not a inclusion of all kind of ancestors. So where all some descendants have been included, but not all descendants are a part of it. Like this was an ancestor, it has two descendants, number 2, and it has another descendant that became 3, 4 and 5. So when you are choosing this group including 1 and 2, you are including descendant 2 of this ancestor, descendant 1 of this ancestor, but you are not including all the descendants of this ancestor. So when you are not including all the descendants, you are including a part of phylogenetic tree. In that situation, that will be called as paraphyletic group. So these were some terminologies that are often included when we talk about phylogenetics: monophyletic group, polyphyletic group, paraphyletic group, nodes, leaves, terminal taxa. You should be aware about these terms. Now, one more thing, that most of the times when you go for reading different literature, when you see trees at many places, you should not be misled by their appearance, because in trees, appearance is not more important. Important is that how they are connected with each other. So if you talk about these three type of trees, you can say that A has emerged from an ancestor, B has emerged from an ancestor, and C and D have emerged from a common ancestor. Can you observe that point? Now, if I draw the same tree in a way like this, so still you can see that C and D have emerged from a common ancestor, and B has emerged from another ancestor, which is common to both C and D. So this is one clade, and this is B, this is A. So the ancestor from which B emerged, from the same ancestor, both C and D emerged, right? So C and D had an ancestor, and their ancestor had common ancestor with B, and then A was having another common ancestor. So if you look at this graph or this graph, they have same meaning. They are identical. That's why I'm saying you should not be misled by their appearances. They do not have any difference at all. Similarly, if you draw the same phylogenetic tree in this manner, you can realize that A is here and you can see C and D here. So if C and D are here and they have a common point from which they emerged out, you can still say that they had a common ancestor, and then this point is connected to this point, from where you can see that A emerged and B emerged, and then you had another common ancestor here. So it does not make any difference whether you draw a tree like this, or you draw a tree like this, or you draw a tree like this. More important is how leaves are connected to a branch. It's like when there is a plant outside and when wind is blowing, then the shape of those branches or the position of those branches may be different. It may not be constant, but the position at which those branches are connected will always be same. So this is how you can connect yourself with phylogenetic trees. If you pick up a point or leaf or branch of a plant and stretch it somewhere and place it somewhere else, it does not make any difference to the phylogenetic tree unless you cut a branch and then you glue it somewhere else. Then it is going to give different type of tree. So if you move branches anywhere, that's not going to make any difference unless the size of the branches or length of the branches is representing some distance. So I'm not going into that right now. Here, the length of branch does not represent any kind of distance, and in that situation, any shape can be acquired by a tree. Similarly, you can also see another example. This particular example also says that A, B, C, D, E, these are 5 different leaves or 5 different taxons, and they are connected in different ways. And you can draw different type of trees based on same information. So all these 3 trees are again identical. They don't have much difference. All right. So this is something I want to say that you must have to remember with caution, that don't get misled by appearance of the trees. You always have to look connections from where the branches are appearing. That's very, very important in case of phylogenetic analysis. Now let's go back and look at some of the more textbook terms to make it more clear. You will hear a lot of textbook terms while you do phylogenetic analysis. One of them is called as dendrogram. So dendrogram is a kind of general term which includes phylograms, cladograms. So when you are doing phylogenetic analysis, whether you are considering evolution or not, anything that looks like a phylogenetic tree can always be called as a dendrogram. Whereas cladogram is something that represent branching pattern. That is, the branch length do not represent time or relevant amount or character change, but they represent descent. So what a cladogram is? Cladogram is a type of dendrogram that represent descent or ancestry of particular taxon. Now another term that you know is chronogram. So if you have a phylogenetic tree that explicitly represent time scale, means number of years that have elapsed between 2 taxons, and that information can come from various things. It can come from carbon dating, it can come from the heuristic approach of molecular changes like changes in base pairs. So by any means, if you know the distance between 2 taxons, the time-based distance, and then you place that time onto the vertices or lines, then you are going to call that as chronogram. And phylogram is something which is phylogenetic tree, where branch length is proportional to the amount of character change. So character change means it may not always be like time. If you are making a tree of DNA sequences, so the length of those branches, they will be proportional to divergence, or you can say number of times the base pair has changed. So suppose we looked at the first example in the slide, different names, Amit Kumar, Sumit Kumar, and different names. So you can say if I want to create a cladogram out of it, it's very simple. So what I made was cladogram. The length of branches did not have any sense there. But if I know the difference in the percentage difference in the sequences, like if I know Amit Kumar and Sumit Kumar has 80% match, Sumit Kumar and Aditya Arya has 10% match, and if I use that character matching or percentage of characters and I make the lengths of trees or branches in accordance with that, then that tree will be called as phylogram. So do you understand? Phylogram will have characters proportional to the length. You can say chronogram is a special type of phylogram where character is time, right? And cladogram is something which doesn't have anything represented by length of the branches. And altogether, any kind of tree, whether phylogram, cladogram, or chronogram, they together can be given a general name that is known as dendrogram. So does that make sense to you? These terms are clear whenever you hear most of the times? And particularly when we are right now discussing about multiple sequence alignment, since we have discussed about 16s and we are looking at the phylogenetic relation. So obviously the kind of tree that we are going to develop in the end will be a phylogenetic tree or phylogram. And in that case, vertices will have a sense of character change. Besides these things, there are a few more newly emerging terms. One of them is known as the Dalgranogram, and Dalgranogram is somewhere like a transverse section of a tree. So if you make a tree and if you take a transverse section and the thickness of branches are also representing some of the parameter, then that kind of phylogenetic tree is known as Dalanogram. It was recently represented in some of the literature. Then next one, another kind of cladogram is known as phylogenetic network. So this is a much broader term when you have one phylogenetic tree connected with another phylogenetic tree, and then there are many phylogenetic trees which are joined together. Then they together make a very, very complex network, and that's known as phylogenetic network. So phylogenetic network is, you can say, collection of many phylograms. So when you connect many phylograms together, like thousands of phylograms together, then that kind of network is known as phylogenetic network. Besides this, there are a few more terms like spindle diagram. They were developed by Alfred Romer, and they are also called as Romarograms. So Romarograms are slightly different phylogenetic trees, which on their vertices have geological time. That means they are representing the overall evolutionary process, particularly in geological time scale, like million years ago and billion years ago that you study in geology. And that kind of tree is known as Romarogram or spindle diagram. So you can see here such kind of diagrams you would have seen. This is Romarogram. As you can see, these are the epochs and era which are representing geological time scale. So you will always have geological time scale on vertical axis, and then these species will be connected together. So you can connect that in which time period, which type of species originated. And something that Amit also shown you in one of the previous slide is known as coral of life. So when you connect all the species in the world together and you prepare a phylogenetic tree, and it will be very, very complex in the form of a spindle, that is known as coral of life. So this is how we come to an end of various type of terminologies. You can find these terms very easily, and we have made a provision to share these presentations as well. In the evening, we will see how we can share these presentations. Now, another thing, when you talk about the phylograms, that was textbook information, but there are a few more terms that are used to classify phylograms. A variety of phylograms will be observed. One of the criteria is to classify them on the basis of whether they have a root or not. Just like you can see a plant, if plant is live, it can have root, but if you cut the plant, it will still be plant, will have branches, will have leaves, and it will have no roots. So if a plant doesn't have a root, in that situation, you can say that origin of that plant cannot be deciphered. If you cut a plant and take it to your home, you will not know where it was planted. But the point of time when it is having roots, you can find out that where it was planted. So on that basis, a tree phylogram can be called as rooted or unrooted. As you can see here, there are two type of trees. The basic difference between rooted and unrooted tree lies somewhere at the oldest ancestor. Okay, so the moment of a relatively more clear picture of this thing can be drawn like this. In most of the textbooks, you will find the rooted and unrooted trees being represented like this. So let us say there are 4 taxons A, B, C, and D, and they are connected to an old ancestor called E. Then you can say you know the origin of A, B, C, D, from where they originated. You know their ancestor. But if I make a tree like this, I have A, B, C, and D, and I do not know that from where they originated, what was their original ancestor. So the only thing that I need to do to make this rooted is to know about an ancestor. The moment when I know that from where C and D originated, from where A and B originated, I can pull this down and I can get a tree that will look like this. So you can introduce a root by knowing the oldest common ancestor. So the only difference, what you can note down or what you can remember about rooted and unrooted tree is when you know the oldest ancestor of the tree, then it is called rooted, and when it is not known, then it is called as unrooted. So we can have both these things, and in fact, we might have various algorithms. There are mathematical formula which are used to determine the total number of trees which are rooted and unrooted based on how many leaves you have, how many taxons you have. So I'm not going into a lot of mathematics out of it. You can go through standard textbooks and can come to know if there are 3 taxons, there will be 3 rooted trees and there will be 1 unrooted tree. If there are 4 taxons, there will be 15 rooted trees and 3 unrooted trees. But if you go to higher number like 5 taxons, 105 rooted trees, 15 unrooted trees, and maybe even higher when you have 7 taxons, there will be 10,395 rooted trees and 954 unrooted trees. So you can see that as it is related to the binomial relationship and it is related with the factorial function. So on increasing the number of taxons, the number of possibilities of various type of trees are increasing exponentially. Now, another criteria to group them is on the basis of how many branches does a tree have. So right now what we saw in a tree that was given or already shown to you, there may be trees which are called as bifurcating. Now why they are called bifurcating? Because at every junction, this is a node, you are seeing 2 branches coming out. Branch 1, branch 2. At this junction, you still have branch 1, branch 2. So if you have a tree where you have only 2 type of branches, then it will always be called as bifurcating tree. But in some instances, if let us say from one node, there are 3 branches emerging out or more than 2 branches emerging out, then that tree will be termed as multiplicating tree. So this is another way of classifying phylogenetic trees. They may be bifurcating, they may be multifurcating. Another term that is often used in the trees is labeled and unlabeled. So it's very, very intuitive and simple. Everybody can understand. If there are labels on a tree, that means the taxons or phylogenetic groups, they have been marked, then it is called as labeled tree. If they have not been marked, it will be called as unlabeled tree. So right now, I have shown you both the trees are unlabeled. And if I place values onto it, like 1, 2, 3, 4, I make this tree labeled. And there are some mathematical relationships, just like rooted and unrooted trees can be calculated. We can also calculate the number of labeled and unlabeled trees, because number of unlabeled trees will be very few. Unlabeled trees will just tell you about the topology. They will just tell you about topology. You do not know whether this is 1, this is 2, this is 3 or 4, but there can be labeling done in multiple ways. So did that make sense to you? Is that clear, that we can classify phylogenetic trees or phylograms on different criteria? They can be rooted, unrooted, they can be bifurcating, multifurcating, they can be labeled or unlabeled.